Hi, my name is Goldis Malek. I am an associate professor at Duke University in Durham, North Carolina. I have appointments in Department of Ophthalmology and Department of Pathology. And since I was a grad student, we've been interested in trying to understand the pathobiology of retinal blinding diseases, specifically age-related macular degeneration. And I started with trying to learn about the pathology, and then I moved on to trying to create animal models. And what we're doing now is to try and identify pathways that contribute to the pathology. And if we can find potential molecules that are involved in creating that pathology, then we can target it and hopefully either reduce it or maybe even prevent it, ideally prevent it. So some of the targets we're looking at are a large family of transcription factors. And essentially what they do is they regulate expression of genes. And these transcription factors are called nuclear receptors. There are a total of 48. And basically, we've done a study where we identified candidate receptors that we should look at in detail. And what I presented here today was specifically about one receptor that regulates lipid metabolism and inflammation. And these are two pathways that contribute to AMD progression. And what we did was basically look at the expression of this receptor in the cells that are vulnerable in AMD, and these include epithelial cells that are essentially nurse cells to the retina, as well as the outer vasculature, which basically provide blood and energy to the retina. After that, we looked at what happens if you take that out of a mouse. What happens to the eye? Do we see any pathology that's similar to what you see in humans? And indeed we do. We see very characteristic lesions called drusen in the mouse. They're sub-RPE deposits, so they're below the RPE deposits. They contain lots of lipids. We see an increase in inflammation. So we think that knocking out the receptor gave us a really nice animal model to study the disease more. And the final thing I presented was basically we can go in, we can target this receptor, try and activate it, and by activating it, improve the health of AMD vulnerable cells, so the epithelial cells and the choroidal endothelial cells, the nurse cells to the retina and the outer blood supply. And so essentially what we try to do is we start from cells, we go to the tissue itself as a complex, and then finally we try and see if we can do therapy.
What are your next steps in this line of research?
The most important thing is once we find the target, we want to identify a drug that has the least amount of toxicity and the greatest bioavailability. So we're working with a small molecule designers, and what they do is they look at the molecule that we are interested, our drug, they make modifications, and we test those out in animal models to make sure there's little toxicity and that it actually does what we want it to do, which is to remove those deposits and improve the health of the retina and the outer blood supply. Our long term, of course, is we have, once again, this large family of transcription factors, and we've identified all these different candidates. So we try and apply the same paradigm, which is to first decide, does this particular receptor play a role? Can it improve the health of the cell? And then finally, can we target it? So we're strategically going through these receptors and trying to understand their biology and their potential for therapy.
What type of drug delivery system are you looking at using for potential therapy?
Ideally, a lot of people are interested in eye drops, and I think that if it comes to it and patient compliance is high, then eye drops would be ideal. Right now, we're doing pure systemic administration, but in the future it would be great that if indeed eye drop compliance is not high, is that we could create something that would allow for slow release with an injection in the eye. But that's long term down the line. Right now, for our purposes, we're doing pure systemic delivery to make sure that it actually gets to the eye and gets to all the other places that we need it to go.
How long before you'll be ready to move into the next phase of this research?
So right now, the small molecule company is actually making the drug for us. So as soon as we get that, we'll be able to test it in our animal models. And it just depends on making sure that the drug is the right drug. So if it comes back and it needs to be tweaked, we'll have to tweak it a little bit more, and we'll continue to do that before we go any further beyond that.
Is there somewhere people could follow your research if they're interested?
They can go to the Duke Eye Center website. I have a website there. I also have a website through Duke University Department of Pathology, so Department of Ophthalmology and Department of Pathology, and our papers are available on PubMed. So.